For about 100 years, the textbook said the most expensive organ in your body had no garbage system at all. They were wrong. It has one. It just refuses to run while you're awake. Here's the problem the brain has, and it is a real engineering problem. Your brain burns roughly a fifth of all the energy your body uses, packed into something the size of 2 fists. Burning that much fuel produces waste. Broken proteins, spent metabolites, cellular debris. Every other organ you own has a way to drain that waste away. They sit in a bath of lymph, a second circulatory system whose entire job is collecting the trash that blood leaves behind and carrying it off to be destroyed. The brain does not have lymphatic vessels running through its tissue. Open an anatomy book from the year 2000 and it will tell you flatly that the brain has no lymphatic drainage. So for a century, the assumption was that the dirtiest, busiest organ in the body somehow kept itself clean by slow diffusion alone. Waste just sort of drifted out. Nobody could quite explain how. In 2012, a team at the University of Rochester led by Jeffrey Eliff and Macon Nedergaard published a paper in Science Translational Medicine that found the missing system. It had been hiding in plain sight, wrapped around the blood vessels themselves. And it came with a catch nobody expected. It barely works while you're conscious. By the end of this, you'll know exactly how your brain takes out its own trash, which single molecule the whole system depends on, and why the cleaning crew only shows up after you lose consciousness. You'll learn what specific piece of garbage gets left behind when you skip deep sleep. And you'll find out why that residue is the same protein that builds the plaques of Alzheimer's disease. There is something your brain refuses to do while you're awake, and the residue it leaves looks exactly like the opening chapter of a disease. Hold on to that. We'll come back to it. So let's go through the plumbing first. Start with where the cleaning fluid comes from. Your brain floats in cerebrospinal fluid. Most people picture it as a cushion, a shock absorber, and it is that. But Eliff's team in 2012 showed it is also the cleaning solution. Using a technique that let them watch fluorescent tracers move through the living mouse brain in real time, they found that cerebrospinal fluid does not just sit on the surface. It gets pumped down into the brain tissue itself, along the outside of the arteries that dive in to feed it. Picture a pipe inside a slightly larger pipe. The artery is the inner pipe, carrying blood. Around it is a thin sleeve of space. The pulse of each heartbeat squeezes that artery and drives clean cerebrospinal fluid down the sleeve, deep into the tissue. There, it washes sideways through the brain, picks up the waste floating between the cells, and carries it back out along the veins. Fluid in along the arteries, dirty fluid out along the veins. A complete wash cycle, with the beating of your own arteries as the pump. Now here is the part that decides everything. That sideways flush, the step where clean fluid actually crosses the brain tissue and collects the garbage, depends on a single molecule. Its name is aquaporin 4. It is a water channel, a tiny pore that lets water pass through a cell membrane, and it sits in dense arrays on the end feet of your astrocytes. Astrocytes are the star-shaped support cells of the brain, and their feet wrap tightly around those paravascular sleeves. Aquaporin 4 is the gate that lets the cleaning fluid move from the sleeve into the tissue. Eliff's team did the clean experiment. They took mice engineered to lack aquaporin 4 and measured how well those brains cleared injected amyloid-beta, the waste protein we'll keep returning to. Clearance dropped by roughly 70%. One missing water channel, and the brain's garbage system nearly stops. A single molecule decides whether your brain gets cleaned at all. Remember that. Damage it, and 2/3 of the disposal capacity vanishes. We are going to find out what happens to the trash that doesn't leave. Macon Nedergaard gave this whole apparatus a name because it behaves like the lymphatic system but runs on the glial cells, the astrocytes. She called it the glymphatic system. Glia plus lymphatic. It was the missing drainage the textbooks swore didn't exist. So the pipes exist. Cerebrospinal fluid in along the arteries, waste out along the veins, gated by aquaporin 4. The obvious next question is when? When does this thing actually run? In 2013, the same lab, with Lulu C as lead author, published the answer in Science, and it reframed what sleep is for. They watched the glymphatic system work in mice that were awake and in mice that were asleep, and the difference was not subtle. During sleep, the space between the brain cells opened up. Not metaphorically, physically. The interstitial space, the gap where waste collects, expanded by around 60%. A wider gap means fluid moves through it far more easily, the way water runs faster through a wide channel than a narrow one. And the clearance rate of amyloid-beta roughly doubled in the sleeping brain compared to the waking one. Think about what that means. While you are awake, your brain cells are swollen close together, doing their work, and the cleaning fluid can barely squeeze between them. The trash accumulates all day. Then you fall asleep. The cells shrink back, the channels open, and the wash cycle finally runs at full pressure. The cleaning crew only comes when you've left the building. This is why Z's paper landed so hard. For all of history, we'd known that every animal sleeps, that sleep is so dangerous and so non-negotiable that evolution kept it, even though it leaves you blind and helpless for hours every night. And we never had a clean answer for what it bought. Here was one. Sleep is, in part, the maintenance window. It is when the brain physically reconfigures itself to be cleaned. Stay awake, and the maintenance window never opens. The fluid never gets its wide channels. The garbage that should have drained out simply stays. And now, I owe you the obvious worry. If this whole thing was shown in mice, how do we know it happens in a human skull, which we cannot exactly open up and watch with a microscope? That question stood for 6 years. In 2019, somebody answered it with a scanner. Here's the open loop I want to close. In 2019, a team caught the cleaning fluid moving inside a living human brain, but only after something electrical happened first. I'll tell you what came first. The work came out of Boston University, led by Nina Fultz, published in Science. They put people inside an MRI scanner and let them fall asleep, while simultaneously recording the electrical activity of the brain with EEG and the movement of cerebrospinal fluid with a fast imaging sequence. 3 signals at once: brainwaves, blood, and fluid. When the volunteers dropped into deep non-REM sleep, a rhythm appeared that does not exist when you're awake. First came the slow waves. These are the large, sweeping electrical oscillations of deep sleep, the slowest brain waves you produce, rolling across the cortex roughly once a second. Then, a few seconds behind each wave, the blood volume in the brain dropped. And into the space the blood vacated, a large pulse of cerebrospinal fluid washed in. They could measure the timing. The peak of the slow electrical wave came about 6.5 seconds before the peak of the fluid wave. The brain waves were leading, the fluid was following. It was as if every slow wave of deep sleep was one stroke of a pump, pulling clean fluid in. That is what came first. The electrical slow wave of deep sleep is the trigger. No slow waves, no pulse. And here is the quiet, heavy implication that runs underneath the rest of this video. The cleaning is not powered by sleep in general. It is powered specifically by the deep, slow wave stage of sleep. The exact stage that thins out first when you sleep badly, when you drink, when you age, when you take certain medications. The fragile stage, the one most people are quietly losing. Now we have the full machine. Pipes around the arteries, aquaporin 4 is the gate, the interstitial space opening in sleep, and the slow waves of deep sleep acting as the pump. The only thing left is to ask what exactly is in the trash. I've been saying the word amyloid-beta and asking you to hold it. Now we open the bag. Amyloid-beta is a short protein fragment. It is produced constantly, all day, as a normal byproduct of the electrical activity of your neurons. The more your neurons fire, the more of it they make. It is ordinary metabolic exhaust. In a healthy brain, it is produced during waking hours and cleared during sleep, and the balance holds. The problem with amyloid beta is what it does when it isn't cleared. It is sticky. Left to accumulate, the loose fragments begin to clump, first into small, soluble clusters, then into the dense, insoluble plaques that pathologists find packed between the neurons of people who died with Alzheimer's disease. Those plaques are one of the two defining lesions of the disease. For decades, the central question in Alzheimer's research has been why amyloid beta builds up in the first place. Iliff's 2012 paper put amyloid beta directly in the experiment. It was one of the solutes the glymphatic system clears, and its clearance collapsed when aquaporin 4 was knocked out. Z's 2013 paper measured amyloid beta clearance specifically and found it roughly doubling during sleep. So the system we've been mapping is not clearing some abstract debris. One of the exact things it flushes out at night is the protein that, left to pile up, becomes Alzheimer's plaque. Put the pieces in a row. Amyloid beta is made while you're awake. It's cleared while you're in deep sleep. The clearing runs on slow waves, so a brain that gets too little deep slow wave sleep produces a full day's worth of this sticky protein and then never fully opens the drain. That is the mechanism on paper in animals. The honest scientist's next sentence is the one that matters. Does any of this actually happen in a living human being who simply slept badly? Because a mouse with its skull open under a microscope is one thing, a person who pulled an all-nighter is another. This is where the work moves from elegant theory to something you should genuinely lose sleep over. Or rather, not. Here is where the story stops being about mice. In 2018, a team at the National Institutes of Health, led by Shokri-Kojori, with the neuroscientist Nora Volkow as senior author, published in the Proceedings of the National Academy of Sciences. They took healthy adults and scanned their brains with PET imaging, which can tag and measure amyloid beta in a living person. They scanned each person once after a normal night of sleep and again after a single night of total sleep deprivation. One night. After that one sleepless night, amyloid beta had measurably risen in the participants' brains. The increase showed up in the hippocampus and the thalamus, regions that are among the first to be hit in Alzheimer's disease. One night without sleep, and the Alzheimer's protein climbed in exactly the places the disease likes to start, in living human heads. But you could object. Total sleep deprivation is extreme. Most people aren't pulling all-nighters. They're just sleeping shallow and broken. Does losing the deep stage specifically matter? Or do you just need hours on the pillow? In 2017, a team at Washington University in St. Louis, with Yoel Ju leading and David Holtzman as senior author, answered exactly that question in the journal Brain. This is the elegant one. They let healthy middle-aged volunteers sleep a full night, the normal number of hours. But every time the volunteers' EEG showed them slipping into deep, slow-wave sleep, a sound played through headphones, just loud enough to pull them out of the deep stage without fully waking them. Same total sleep time. Only the deep part was sabotaged. The next morning, the amyloid beta in their cerebrospinal fluid had gone up. And when the disruption was sustained over several nights, a second protein rose too. Tau. The other defining lesion of Alzheimer's disease. So it isn't just hours in bed, it is the deep, slow-wave stage specifically, the exact stage Fultz showed was driving the cleaning pulse that keeps the amyloid in check. Lose that stage, keep all your hours, and the protein still climbs. If you find this kind of thing worth knowing, the kind of biology that was hidden in plain sight for a century, this channel is built for it. Subscribe, and the next investigation finds you. Now I have to tell you the part that turns this from a worry into something genuinely dark, because so far it sounds like a one-way street. Sleep badly, amyloid builds. If that were all, you could fix it by simply sleeping better, and the story would end cleanly. It does not end cleanly. In 2015, Bryce Mander and the sleep scientist Matthew Walker, working at Berkeley, published in Nature Neuroscience. They studied older adults who were still cognitively normal, and they measured three things: how much amyloid had already accumulated in the brain, how strong the person's deep, slow waves were at night, and how well they remembered things the next morning. The amyloid load predicted the loss of slow waves. The more amyloid a person had built up, the weaker their deep sleep became, and the weaker the deep sleep, the worse the overnight memory. Sit with the shape of that. Poor deep sleep lets amyloid accumulate. We saw that in Jew and in Shokri-Kojori. But Mander and Walker showed the arrow also points the other way. Accumulated amyloid corrodes the slow waves, and the slow waves are the pump that clears the amyloid. So it is a loop. A little amyloid weakens your deep sleep. Weaker deep sleep clears less amyloid. More amyloid builds, which weakens the deep sleep further. The system designed to protect you from the disease is the first thing the early disease quietly switches off so that it can build faster. It is a feedback loop that, once it tips, helps to drive itself. This is the honest version of the headline. Skipping deep sleep doesn't simply make you tired. At the level of the protein, in the regions the disease targets, it makes your brain do the thing the early disease does. It is not a metaphor that poor sleep looks like Alzheimer's. The amyloid rises in the same places, the slow waves weaken the same way, and each one drives the other. A bad night is a small rehearsal. A bad decade is something else. I owe you the limits here, because this channel does not sell you fear it can't back. Most of the deepest mechanism, the pipes and the aquaporin gate, is rodent work. The human studies are imaging and biomarkers, mostly short-term or correlational. No one has shown that fixing your sleep prevents Alzheimer's. And the word glymphatic is still argued over by serious scientists who disagree about the exact plumbing. What is solid is this: sleep, and specifically deep sleep, clears metabolic waste from the brain. That waste includes the Alzheimer's protein, and disrupting deep sleep raises that protein in living people. Everything past that is the frontier. So, if the cleaning only runs in deep sleep, and you can't run it by force of will, is there anything you can actually do? There is one lever, and it is not the one the supplement aisle sells you. The lever is protecting slow-wave sleep itself, not just hours in bed, the deep stage. And the things that wreck it are unglamorous and familiar. Alcohol before bed suppresses slow-wave sleep even when it makes you fall asleep faster. Going to bed at a different time every night fragments it. A warm, bright, noisy room thins it. Deep sleep is also the stage that shrinks most steeply as we age, which may be one reason Alzheimer's risk climbs with age in the first place. The protection of consistent timing, a cool, dark room, and laying off the late drink isn't wellness advice. It is, mechanistically, glymphatic maintenance. There is one more thread, still early, that I find quietly striking. Some of the animal work, including from Nedergaard's group, suggests the glymphatic system clears better when you sleep on your side rather than flat on your back, possibly because of how cerebrospinal fluid drains under gravity. It is far from settled in humans, but it raises the strange possibility that the position you've slept in your whole life has been silently helping or hindering the nightly wash. We don't know yet. That's an honest we don't know. Step all the way back. For 100 years, we believed the brain had no way to take out its trash. It does. It built the drainage into the spaces around its own arteries, gated it behind a single water channel, and then, for reasons evolution understood before we did, it locked the whole system to the one state in which you are blind, deaf, and unconscious. The cleaning crew only comes when you've left the building. Stay in it every night, refuse the deep stage, and the trash never leaves. And the residue it leaves behind is not nothing. It is the first material of a disease that erases people from the inside. There is one axis I left out, and it's the next investigation. We've talked about clearing waste, But the same slow waves that drive the cleaning also drive memory. The night isn't only when the brain is washed, it's when the day is filed. What gets saved, what gets thrown away, and who decides while you sleep. That's the one I'm chasing next. For now, the next time you trade an hour of deep sleep for one more episode, understand what the trade actually is. You are not borrowing energy from tomorrow. You are skipping a wash cycle in the only organ you can't replace. Your brain has been quietly asking you for the maintenance window every single night of your life. The hard part is finally giving it. If you want the next one when it lands, subscribe.